I welcome you all for today's presentation on drug discovery. Myself, Dr. S.A. Kumar, working as scientist in the Division of Pathology, Indian Council of Agriculture Research, Indian Veterinary Research Institute. Let's begin the presentation. Drug development is the process of bringing a novel drug from bench to bedside. It can take 10 to 15 years for a drug to be designed, developed and approved for use in patients. In some circumstances, the drug development and approval process can be expedited. For example, if the drug is the first available treatment for a condition, or it shows a significant benefit over existing drugs. Before a drug can reach a patient, it must go through rigorous testing to determine whether it is safe, effective at treating the condition it was developed for, and to ascertain the correct dosage and appropriate administration route. Drug development can be described as the process of identifying chemical entities that have the potential to become therapeutic agents. The continuing need for drug discovery exists because there are still many diseases inadequately served by existing therapies. Each year sees a couple of dozen new drugs licensed for use, but in their wake there will be tens of thousands of candidate drugs that fell by the wayside. The research and development journey of those new drugs that make it to market will have taken around 12 years and cost around £1.15. The journey will have begun in a university laboratory where researchers, with grants from the research bodies or the pharmaceutical industry, have undertaken basic research to understand the processes behind a disease, often at a cellular or molecular level. It is through better understanding of disease processes and pathways that targets for new treatments are identified. This might be a gene or protein instrumental to the disease process that a new treatment could interfere with, for example, by blocking an essential receptor. Once a potential target has been identified, researchers will then search for a molecule or compound that acts on this target. Historically, researchers have looked to natural compounds from plants, fungi or marine animals to provide the basis for these candidate drugs, but increasingly, scientists are using knowledge gained from the study of genetics and proteins to create new molecules using computers. As many as 10,000 compounds may be considered and whittled down to just 10 to 20 that could theoretically interfere with the disease process. The drug development process involves 5 major steps, viz. Step 1. Discovery and development. Step 2. Preclinical research. Step 3. Clinical research. Step 4. Drug review. Step 5. Post-market drug safety monitoring. Here typically, researchers discover new drugs through new insights into a disease process that allow researchers to design a product to stop or reverse the effects of the disease. Many tests of molecular compounds to find possible beneficial effect against any of a large number of diseases. Existing treatments that have unanticipated effects. New technologies, such as those that provide new ways to target medical products to specific sites within the body or to manipulate genetic material. At this stage in the process, thousands of compounds may be potential candidates for development as a medical treatment. After early testing, however, only a small number of compounds look promising and call for further study. Once researchers identify a promising compound for development, they conduct experiments to gather information on how it is absorbed, distributed, metabolized, and excreted, its potential benefits and mechanisms of action, the best dosage, the best way to give the drug, such as by mouth or injection, side effects or adverse events that can often be referred to as toxicity, how it affects different groups of people, such as by gender, race, or ethnicity, differently, how it interacts with other drugs and treatments, its effectiveness as compared with similar drugs. Before testing a drug in people, researchers must find out whether it has the potential to cause serious harm, also called toxicity. The two types of preclinical research are in vitro and in vivo. FDA requires researchers to use good laboratory practices, GLP, defined in medical product development regulations. For preclinical laboratory studies, the GLP regulations are found in CFR Part 58.1, Good Laboratory Practice for Non-Clinical Laboratory Studies. These regulations set the minimum basic requirements for study conduct, personnel, facilities, equipment, written protocols, operating procedures, study reports, and a system of quality assurance oversight for each study to help assure the safety of the regulated product. Usually, preclinical studies are not very large. However, these studies must provide detailed information on dosing and toxicity levels. After preclinical testing, researchers review their findings and decide whether the drug should be tested in people. While preclinical research answers basic questions about the drug's safety, it is not a substitute for studies of ways the drug will interact with the human body. Clinical research refers to studies or trials that are done in people. As the developers design the clinical study, they will consider what they want to accomplish for each of the different clinical research phases and begin the investigational new drug process, and a process they must go through before clinical research begins. Researchers design clinical trials to answer specific research questions related to a medical product. These trials follow a specific study plan, called a protocol, that is developed by the researcher or manufacturer. Before a clinical trial begins, researchers review prior information about the drug to develop research questions and objectives. Then, they decide who qualifies to participate. Selection criteria: how many people will be part of the study, how long the study will last, whether there will be a control group, and other ways to limit research bias, how the drug will be given to patients and at what dosage, what assessments will be conducted, when, and what data will be collected, how the data will be reviewed and analyzed. Drug developers or sponsors must submit an investigational new drug IND application to FDA before beginning clinical research. In the IND application, developers must include animal study data and toxicity, side effects that cause great harm, data manufacturing information, clinical protocols, study plans for studies to be conducted, and data from any prior human research information about the investigator. Clinical trials follow a typical series from early, small-scale, Phase 1 studies to late-stage, large-scale, Phase 3 studies. Phase 1 involves study participants of 20 to 100 healthy volunteers or people with the disease/condition. Length of study is for several months. Purpose is mainly to ascertain safety and dosage. Approximately 70% of drugs move to the next phase. Phase 2 involves up to several hundred people as study participants with the disease/condition. The length of study is several months to 2 years, with its purpose being to ascertain the efficacy and side effects. Approximately 33% of drugs move to the next phase. Phase 3 involves study participants of 300 to 3,000 volunteers who have the disease or condition. Length of study is between 1 and 4 years, with a purpose to decipher the efficacy and monitoring of adverse reactions. Approximately 25 to 30% of drugs move to the next phase. Phase 4 involves study participants of several thousand volunteers who have the disease/condition. This phase helps in understanding the safety and efficacy. If a drug developer has evidence from its early tests and preclinical and clinical research that a drug is safe and effective for its intended use, the company can file an application to market the drug. The FDA review team thoroughly examines all submitted data on the drug and makes a decision to approve or not to approve it. A new drug application that tells the full story of a drug. Its purpose is to demonstrate that the drug is safe and effective for its intended use in the population studied. A drug developer must include everything about a drug from preclinical data to phase trial data in end. Developers must include reports on all studies, data, and analyses, along with clinical results. Developers must include proposed labeling, safety updates, drug abuse information, patent information, any data from studies that may have been conducted outside the United States, institutional review board compliance information, directions for use. A review once receives and the review team decides if it is complete. If it is not complete, the review team can refuse to file, and if it was complete, the review team has 6 to 10 months to make a decision on whether to approve the drug. The process includes the following. Each member of the review team conduct a full review of his or her section of the application. For example, the medical officer and the statistician review clinical data, while a pharmacologist reviews the data from animal studies. Within each technical discipline represented on the team, there is also a supervisory review. Few inspectors travel to clinical study sites to conduct a routine inspection. The agency looks for evidence of fabrication, manipulation, or withholding of data. The project manager assembles all individual reviews and other documents, such as the inspection report, into an action package. This document becomes the record for a review. The review team issues a recommendation, and a senior FDA official makes a decision. FDA approval in cases where FDA determines that a drug has been shown to be safe and effective for its intended use. It is then necessary to work with the applicant to develop and refine prescribing information. This is referred to as labeling. Labeling accurately and objectively describes the basis for approval and how best to use the drug. Often, though, remaining issues need to be resolved before the drug can be approved for marketing. Sometimes FDA requires the developer to address questions based on existing data. In other cases, FDA requires additional studies. At this point, the developer can decide whether or not to continue further development. If a developer disagrees with the FDA decision, there are mechanisms for formal appeal. Even though clinical trials provide important information on a drug's efficacy and safety, it is impossible to have complete information about the safety of a drug at the time of approval. Despite the rigorous steps in the process of drug development, limitations exist. Therefore, the true picture of a product's safety actually evolves over the months and even years that make up a product's lifetime in the marketplace. FDA reviews reports of problems with prescription and over-the-counter drugs, and can decide to add cautions to the dosage or usage information, as well as other measures for more serious issues. Thank you for your patient listening. Kindly subscribe to my channel for more videos.